Hey everyone, welcome back to another Bio in a Bottle topic review video. I'm Ruben and today we're going to start going over Unit 3 with topics 3.1, 3.2, and 3.3, which all cover enzymes. Enzymes are essential to life. Without them, the speed of biological processes would not have an efficient pace for life to exist. However, what are enzymes? Enzymes are types of proteins in which helps facilitate chemical reactions within our body. Enzymes will bind with certain molecules called substrates at the active site of that enzyme. Each enzyme is specific to their substrate, and they will have a certain shape depending on the chemical reaction taking place. Therefore, enzymes are unable to bind with substrates that have a different shape to them. This shape is very specific, and substrates will bind to enzymes almost like a puzzle piece. When enzymes bind to a specific substrate, the enzyme can change its shape to match the substrate even more perfectly, forming what we call an induced fit. This substrate will not remain binded to the enzyme forever, and therefore the enzyme will either break down the substrate or build it up. Once this is finished, the enzyme can release the changed substrate, which is now called the product, and the enzyme can return to its original shape. Enzymes can be used over and over again, causing them to be called catalysts. Enzymes are so important because they speed up chemical reactions by lowering the activation energy to complete the reaction. The higher the activation energy is, the slower the reaction takes place. Enzymes will decrease the activation energy, causing for the reaction to take place faster. This efficiency and speed allows for life to exist. This efficiency also depends on the amount of substrates within the environment, as the speed of the reaction will speed up if there is more substrates, as a substrate is more likely to bind to the enzyme by chance. An increase in temperature can also assist in the efficiency of a reaction, as the higher the temperature, the faster the reaction. However, enzymes can only get so hot, and enzymes will have limits to how hot it can get. When an enzyme is within extreme high temperatures, the enzyme will denature, which means that its shape will change, causing it to be unable to bind to substrates. Enzymes can also denature when they are within an environment that has a pH outside of the optimal range. When a pH becomes extremely acidic or basic, the enzyme will denature. In some cases, if the temperature and pH go back to normal, the desaturation of the enzyme can be reversed. Next, we will be talking about inhibitors. There are two types of inhibitors, a competitive inhibitor and a non-competitive inhibitor. They both present the substrate from binding to the enzyme, but in different ways. The competitive inhibitor will target the active site of the enzyme, and once it binds to it, the substrate is unable to bind anymore as the space is taken up. The non-competitive inhibitor, on the other hand, will attach to a different part of the enzyme, called the allosteric site, and once it binds to this allosteric site, the shape of the enzyme will change, causing the substrate to be unable to bind to the active site. That's it for this video. Thanks for watching, and make sure to like and subscribe for more AP biology content.